Okay. Okay, well, thanks very much. It's a pleasure to be here, and I'm really looking forward to the day, and I've enjoyed the talk so far. So I'm a neurologist, as it says there, so I know next to bugger all, as well, let's say bugger all, next to nothing against about cancer. So anyone who has to answer cancer questions, keep them to yourselves. But anyway, so what I really want to get across is neurodegeneration is important, and where I think the symmetries may align both biologically and etiologically. But neurodegeneration has been neglected. Funding, I was talking with a colleague earlier this morning here about the lack of funding that's gone to neurodegeneration. And yet more money is spent on dementia than is spent on cancer, cardiovascular and diabetes combined. However, 98 or 99% of that is on care. And it's very expensive to look after people who are dementing because they can't do much for themselves, particularly in the latter stage of the diseases. And so it's a huge, huge burden. And not surprisingly, most developed economies are thinking very hard about how to combat this. They've got some of the costs there. Put 40 billion euros across the EU. Obviously, post-Brexit, we may say, who gives a damn, but that's something else. But the other problem we have for neurodegeneration is that we don't know much about the pathways. So this is an old slide. I've had this for a few years, and you're not meant to be able to read this. Cancer's been a huge success, and those of you who are cancer biologists in the room will know this. You know, in the last 30, 40 years, huge numbers of pathways and complexities of the molecular pathways have emerged, and importantly, many of those steps are now druggable in some way, incompletely perhaps. Neurodegeneration looks a bit like this. It's a bit better. This slide is a few years old to say, but it's a bit better than that now, but it's certainly a lot more patchy. And the idea is how are we going to get our picture for neurodegeneration looking like that? That's part one, and that's really what I've been driven by for the last 10 years. But also, it's all to come up, and I'll talk a bit about this, is actually there's a bit of symmetry, there's a bit of parallel work going on with discoveries in cancer that have insights into neurodegeneration and vice versa. So turning to Parkinson's disease, so I started working on this 20-odd years ago. I was interviewed for a lecture position at UCL, and they said, we want you to work on Parkinson's disease, which I was not very impressed with. It was clearly a non-genetic disease. We all knew it was non-genetic. The epidemiology, the twin studies, the rather small-scale association studies that had been done at that time had all shown, in inverted commas, that it was a non-genetic disease. There were 3 families published in the literature at that time, and neurologists like myself would spend days arguing about whether it was whether it really was Parkinson's disease or not. There was too much dementia, it was too early onset, it was too this, it was too that. And that's where the field was at 20 years ago. However, I'll show you very briefly in a moment, so it's been hugely successful and fruitful for the field to be working in this area. And I think that's been driven by the fact that the phenotype, complicated though it is actually reasonably constrained. So even though it's not a very heritable disease, that if you take the phenotype of tremor, rigidity, bradykinesia, you have a very high prediction of a certain type of pathology, the Lewy body shown on the end of that slide here, and that has a rather limited number of pathways to the disease. And I think we're picking up that limited number of pathways with genetics. So this is a brief summary. There are a few more Parkinson's Mendelian genes than this, but these are the main ones. And what I've done on the right-hand side there in preparation for this talk is say what the role in cancer. I mean, this is a loose, these are mutations that have been imported in cancers and so forth. And you can see there's a lot of them. In fact, none of the major Parkinson's disease are free from some putative role, and it is no more than that at this stage for many of these conditions, many of these proteins in cancer pathogenesis. And that got me thinking a few years ago, and we put up a couple of opinion pieces out about 4 or 5 years ago, saying, can we learn something from the other field? And if, in essence, I wondered whether neurodegeneration could piggyback on some of the success that's happened in cancer, both in terms of molecular pathway description and also molecules to step forward. And this is what has sparked some of our interest. But we have huge problems in neurodegeneration research. Problems that cancer has its own problems, but we have different ones. First up, we have the nervous system, which I won't coin Woody Allen, but it's a very complicated system and important. We have real difficulty with disease definition. I mean, defining cancer may seem complicated enough, but it's not that complicated compared to when you've got, say, dementia. Is it frontotemporal dementia? Is it this type of dementia? Is it that type? Is it Parkinson's? There's huge congresses on all these topics. And not surprisingly, if you've got that diversity, you've got a lot of difficulties in having a coordinated approach to it. I actually favour the mindset that we should be thinking about pathways to disease. We talk about disease phenotypes. This is Parkinson's, this is Alzheimer's, this is whatever, ALS and so forth. But actually we should be thinking more like biologists as clinicians and think about what is it about the molecular pathways that are driving that disease state, and I'll touch on that as I go forward. One of the major problems you have is access to tissues. If you have cancer, you have it removed. That's part of the therapy. If it comes back, you have it removed again. This gives biologists a chance to have tissues at different parts of the evolution of the disease. They get different chances at the treatments, so the tumour's been treated, so they can see the evolution of mutations that emerge in cancers. We don't get anything like that in neurodegeneration. We get the slow, progressive decline, as spoken about by the previous speaker. We do get the brains post-mortem sometimes, but you're looking often at the tombstones. You're looking at the cells that have managed to survive the process of the disease. The cells you're really interested in are the ones that have already gone missing. And so we have problems there that are going to be difficult to combat. And then finally, we have model systems. It is difficult. Working on mouse biology in cancer fields has been very productive indeed. Working on mouse models in neurodegeneration has not been so productive. You have to usually do a huge biological perturbation in order to get something that looks like Parkinson's or Alzheimer's in a mouse. You have to overexpress a protein, massive fold. You have to age it well beyond sort of a normal thing. You have to stress it in some way. You have to give it some sort of noxious poison and so forth. And so we haven't got very good model systems. If I return to the genetics for a second, so this plot in essence was shown in the previous speaker for Alzheimer's disease, that at the left-hand side here you've got things that are rare in the community, often very rare indeed, but when you've got them, they give you a very high chance of disease, and at the far end, at the brown end, you've got things that we've all got, and that we have a selection of these variants that increase our risk of asthma, diabetes, in my case Parkinson's disease and so forth. If we think about that for Parkinson's disease, there's been huge success in mapping Parkinson's disease genetics. These are just some of the major papers that the group that I'm part of have put out the last few years. And as you can see, rather significant numbers. And the map for Parkinson's disease now looks like this, that we've got a number of high penetrant Mendelian factors in the sort of dark grey. We've got in the light grey a bunch of commonly occurring variants, and then we've got somewhere in between. And this has all happened in the last 15 to 20 years. In fact, it's the 20-year anniversary of the first Parkinson's gene being cloned about literally this month, and that's happened in this time. If we start to think about, okay, what's the evidence of a tie-in between these two diseases? Somebody asked the question to the first speaker whether it's just chance, and I think that is still a very fair question, I have to say. You can start to think a bit about some of the epidemiology, and I'm just briefly going to summarize some of the things here. So there is if you've got a risk of colorectal cancer, you've got a decreased risk of Parkinson's disease. This is just one of the fairly recent studies that came out. And these are well designed. I like epidemiologists. They really have thought very deeply and hard about how to design things to minimize the biases that can ensue from just doing purely observational studies. Somebody asked about prostate cancer about half an hour ago. Here's a very large study. Denmark has very good, and Scandinavia generally has very good registries for diseases, and so they do very good population-based structural studies. And again, an overall risk reduction of Parkinson's disease of about 27%. Some nice meta-analysis published by Bajaj with Jenny River involved just a few years ago. And in essence, you can see here that if you have Parkinson's disease, you've got a decreased risk of cancer and vice versa. there is an exception to that, and the striking exception is melanoma, that there's repeated studies show that if melanoma and Parkinson's have a positive correlation, again shown in this meta-analysis. If you look at the bottom diamonds there, the overall risk's about 1.5 stage risk of the disease. So modest, but significant over very large studies. Okay, so we've got these observations. We've got overall decreased risk for Parkinson's disease and cancer, negative correlation, and you've got one example of a positive correlation. So we can ask a question with combining genetics and risk and say, what about melanoma genetics? What do we know about melanoma genetics, and does any of that light up when we now look at these rather deep data sets that we have of Parkinson's disease? And the answer is not a lot at the moment, even with this resolution, even with this scale of activity that we've got. So MC1R is a polymorphism associated with risk of melanoma, but now there was a report in Annals of Neurology a couple of years ago showing that it was a risk of Parkinson's disease. A larger scale secondary study by the International Parkinson's Disease Genetic Consortium did not confirm that, in fact showed complete unity, so we have to wait and see who's right on that. You then can take a rare variant view. You can say, okay, well, let's see if there are any disease Mendelian type mutations, level of mutations coming out in melanoma genes. And the answer of the resolution of this size of cases and controls is not a lot. But this is rather modest. If the cancer biologists in the room often now can sequence, they've got tens of thousands of cancers or cancer patients have been sequenced. So I think we still have to keep a bit of a health warning on that. But at the moment, we're not seeing a lot. You can do the other way around. You can look at all these Parkinson's genes I've mentioned in passing, and do any of those have any risk of melanoma? And it does appear, as a paper last year, showing that you've got an excess of Parkin deletions, PARK2, the second Parkinson's gene to be found in patients with melanomas. And I'm going to return to this gene in a moment. Okay, so there's circumstantial evidence at best with those bits of data. But can we think of other areas where there's some molecular commonality between the two diseases? And I think the striking one is some of the recessive ataxias, and this is a story that's been around for a few years. Interestingly, though, it's only come to the insights of most neurologists in the last few months, in fact. I had a colleague who works in this area, came to me with the news and views from Nature saying, look at this, DNA repair is important for neurodegeneration. And I said, well, yeah, but we've known about this for some of these genes for a while. So it may be well known to the cancer guys in the room, but to neurodegeneration, less so. So the most well-known one, of course, is ataxia telangiectasia. This is an early onset, really unpleasant disease. It gives children this balance disturbance due to pathology in the cerebellum. But it's a multi-system disorder. It affects their nerves, so they get a neuropathy as well. They get telangiectasia, shown here in the eyes. They sort of look like it's not really bloodshot. It's like this spindly red blood vessel. But they get humoral immunity problems, and they get cancer risk as well. In fact, their parents, the heterozygote carriers, obligate carriers, are also at slight increased risk of cancer as well. And this has been known about for 15 or more years. There are some other diseases that look much rarer than AT is rare enough. Some other rarer diseases, Nijmegen breakage syndrome and so forth. And these all involved in double-strand DNA breaks. And it can't be by chance, talking about networks or a little bit of a sort of network, if you like, a molecular network, that these are so closely aligned in the same pathological molecular process. So this surely indicates, I think, that double-stranded DNA breaks are obviously important for cancer. That's been known about for many years and researched by many groups. But also, it must be telling us something about the pathogenesis of cerebellar neuron fallout and other parts of the nervous system as well. DNA single-stranded breaks, this is a more recent discovery in the neurological field, at least, with AOA1 and 2 coming through and so forth. And then just a couple of months ago, a paper in Nature came out showing that mutations in this gene XRCC1 show this is a loss of function, these mutations, recessive mutations, when this family, this case in fact, had loss of this protein, they activated PARP and had problems with single-strand breaks. I was a reviewer for this paper. I think the other reviewers were the guys who wrote the News and Views, so I think we now know who they were. But anyway, and the biology was beautiful. I mean, it really is a really impressive piece of work. Genetics, there was one case. If this had gone to a good standard genetic journal, it would not have been accepted, because finding one case, albeit with mutations which were likely to be pathogenic, with great biology, is persuasive. We know now with very large-scale sequencing, we've all got stuff bad in our genomes. So it was not, not entirely genetically proven. We haven't published it yet, but we've looked now in our cases, and we've found this mutation, this one here. We found this in 2 more cases, homozygously. So I think that they were right. I thought they were right at the time, but I think this shows that they're right. So this mutation is turning out to be this gene is giving important insights. You can also use genetics to inform on interactions between sort of hardcore Mendelian genetics and GWAS signals, and I want to give an illustration of that, which I think has been instructed in the last 9 or 12 months or so, maybe 2 years. So the polyglutamine disease is probably hopefully well known, certainly well known in neurological circles. There's a family of these disorders. They're all due to abnormal repeat lengths of CAG in the coding bits of the gene. They encode glutamine, and so known as polyglutamine diseases. And you're allowed, it varies from gene to gene, you're allowed about 20 repeats or 20 glutamines, and that's okay. You get 37, 40, depends from disease to disease. That's bad, and you get this often, nearly always, fully penetrant disease. So you can have 35 polyglutamines be normal, have 38 glutamines, and you get this disease. A very tight switch between normality and pathogenesis. And they cause Huntington's disease and a bunch of ataxias. I went to a Gordon conference 10 years ago, and I was giving a talk on the ataxias, actually, to mostly an HD group. And I was just saying, what you guys want to be doing is you want to be doing a GWAS of the variants that's not explained by the CAG repeat diseases. And they said, absolute waste of money. We don't want to be doing that at all. We know what most of the variance is. We know what the gene that causes disease. If you either have this mutation, you get the disease. If you don't have it, you don't get the disease, and we're not going to be interested in it. So I didn't do anything with it. So all I had was the idea. But we've known for a while that if you have the repeat length, it predicts the number of repeat diseases. But then a very nice paper a year and a half ago came out from Jim Drusella and others at Harvard and the big genetic consortium, showing that when they did do a very large-scale GWAS for the bit of the variance of the age of onset that was not explained by the CAG repeat. About 70% is explained by the CAG repeat length. The other 30%, they did a large-scale GWAS. They showed a number of gene regions, most prominently 2 on chromosome 15 and 1 on chromosome 8. They did some network analysis. I don't know how much this would impress our first speaker. See, it's not floating ground. Could you help me with my slides? No, I lifted this from their paper, but the message is, apart from my crappy slides, is it was kind of persuasive that actually a rather simple idea came out, and it was to do with DNA repair again. And you've got a repeating mutation here. So this had a kind of believability about it. We'll have to see whether it's right. So a follow-up group, this is something I had a small part in, involved in, published last year, looked at the other scars, looked at the other CAG repeats, because it seemed to me a pretty obvious question really is, okay, is the variance that you've explained with these GWAS hits explaining just HD, or does it explain other CAG repeat length problems? And so it's a simple question. So we took the sort of 22 top SNPs from the first paper, applied them to a much smaller number, because these are generally rarer diseases, and you can see you find significant values, and the direction of effect is in the same direction. It's in the opposite direction, we'd have been a lot more worried about it. So it seems there are genetic factors out there that regulate or control in some way the variants of onset for a whole range of polyglutamine diseases. And now you can start to speculate. It is only speculation at this stage as to how this might cause disease risk, whether it's something to do with somatic mutation. Is it the fact that if you've got DNA repair abnormalities or preponderance in certain ways, that in the brain tissues of these patients, in some of them who've got the risk alleles, they have longer repeats inside the brain than they have in their blood. And that's why they get earlier age of onset, i.e. their biological age is different to their, if you like, their chronological age in this sense? Or is there some other mechanism? I think it's very early days to say more detail on that at the moment. I would give a little word of caution. GWAS are mapping strategies. They tell you where something is in the genome. So if you look at this, this is the... So this is GWAS these are both GWAS hits from Parkinson's disease. And if you, this is chromosome 4, you've got a whole bunch of SNPs, 10 to the minus 25, so a really significant p-value. There's really only one gene in town, alpha-synuclein. If we'd not known about alpha-synuclein from Parkinson's disease from Mendelian genetics, we'd have got it from our GWAS. However, many of them look like this. The top SNP here is 10 to the minus 8, and there's a whole bunch of genes in there, and some that we have not even listed. We do not know which gene's in there. So when you see a mapping saying it's clearly this gene because it fits some biological plausibility, I think you've always got to be a little bit uncertain what's the genetic evidence of the mapping strategy for these things. And that's true for all GWAS, not just to do with what I'm talking about today. If we try to build a sort of understanding of the pathways of Parkinson's disease, to return that for a moment, we can map on some pretty well-established molecular processes now. We've got some protein folding abnormalities, amongst other things. We've got lysosomal biology. We've got mitochondrial biology, all in some hand-wavy way leading to neuronal death. And I just want to pick on these for the moment because these are important for Parkinson's disease. This is something we've worked on for a while, but also it's starting to tie in with the link with cancer, as I'll go on to show you. So there's a lot of work, this is by our group, but there's lots of other groups working on this. But basically, it's got a PINK1, we discovered it in 2004, it's got a mitochondrial targeting domain, so we knew it was going to mitochondria, went on to show that as amongst other groups. And there's a whole range of mitochondrial phenotypes that can be robustly measured and discovered in mouse models, in neuronal cell models, and so forth. And it often to do with calcium influx into the mitochondria and its knock-on effects on reactive oxygen species. So there's a reasonably hardcore biological readout you can obtain from PINK1 derived knockouts. But I've been really struck by there's two papers back-to-back, over 10 years ago now, on Drosophila, and it's sort of shown the importance of some animal models in terms of discovery things. So blue is wild type, pink is knockout, and orange is the rescue. And you can see if you've got the knockout flies, or the cells from the flies, and you poison them with different things, all the cells do badly, and you can rescue it by re-expressing PINK1. Okay, so far so good. But they also did some other rescues. Here is the wild type of the thorax, here's the knockout, and here's the rescue. But this time it's not rescued by PINK1. These flies are still deficient to PINK1. It's rescued by Parkin, and the reverse experiment doesn't work. So they've shown that PINK1 is north of Parkin in the fly, and that they're in the same pathway. Pink and Parkin are both recessive Parkinson's genes. So this was 10 years ago, 11 years ago. The chance that was by chance and not going to be useful to mammalian systems like humans seemed a little unlikely. And over the last 10 years, that's been shown to be true. These genes, Parkinson's genes, are in the same pathway. A third recessive Parkinson's gene was found about 6-7 years ago, FBX07. And it's one of the FBOX proteins, and we wondered where there was going to be a specifying agent for an E3 ubiquitin ligase, and I'm telling you that because it turns out to be true. And so we ended up with a model, we and the field, that looks a bit like this. Here's your mitochondria. we're depolarizing, we're damaging it. It's a highly reactive oxygen species rich environment all the time. So this is slightly damaged mitochondria. Right, I better get a move on. When that happens, you decrease cleavage of PINK1, you increase the non-cleaved, and this acts as signals to recruit parkin, which is a cytosolic protein, which then ubiquitinates mitofusin, which stops mitochondria fusing, so they can't fuse, and so you're pushing it down the degradation pathway. And off it goes. And this is obviously, you can imagine it's being very tightly regulated. To the last few slides of the previous speaker, neurons need to be able to regulate turnover of these mitochondria, otherwise we're going to be in problems. And you can imagine getting pushed towards neurodegeneration quite quickly. Now, the reason this might be important for cancer is shown in this recent paper by just published about 2 weeks ago in Molecular Cell. They looked at a whole bunch of cancers and showed a staggering amount of deletions in Parkin, in a range of cancers. 69% of cholangiocarcinomas carried mutations in Parkin, which sounds a lot. I mean, I don't work in cancer, but that sounds an awful lot to me by any measures. And they showed that if you had abnormalities in PARK2, you had decreased survival as well. And so they gave very strong evidence of this being an important tumor suppressor gene. They went on to speculate how it might work in the PI3 kinase pathway, a pathway that will be well known to the cancer biologists in the room. I'll move on. Okay, so the final few slides to tell you where I think one of the areas we should be thinking about how we can combine fields and make some insights is Mendelian randomisation. So the idea here is that we get our alleles distributed at birth randomly. And those alleles encode proteins, of course, and those proteins do important things for us. And if we can understand what the exposure, what the protein does in terms of the risk of an outcome, we can get some insights into the causality of the disease process. So let's just take something, for example. We know variation in the FTO gene drives, in part, BMI, our weight, and we can ask questions about whether BMI has an impact on Parkinson's disease. It's been well known, I mean, if you're a neurologist, you never see an overweight Parkinson's patient. Never is an exaggeration, but hardly ever. They're always skinny. Now, are they skinny because they've got a terrible disease and disabled and they're sitting still and wasting away? Are they skinny because they're moving around with their tremors and burning calories? Is it part of the illness? Or is there something more causally related? We can ask questions based on genetic predictors of BMI, because there's now been large GWAS done on people who have various weights, and we know how to capture quite a lot of the variation between our BMI. We can say, since they were distributed randomly at birth, we can ask questions about how that drives our absolute BMI. This is a paper that's just been accepted in PLOS Medicine. Should be out in a few weeks, I hope, or a month or so. That basically, if you're chubbier, you've got a decreased risk of Parkinson's disease. And it seems to be driven by the weight itself or the BMI itself, not just genes because of the method. Now, we can use that to think about treatments for cancer and neurodegeneration, which is where I want to end up. We could either do this model, which is sort of guess, or part guess, and that's been a model that's been successful for medicines, some medicines, but as we heard about in the last speaker, it's been terrible for neurodegeneration. We've really done very badly indeed in having new therapies for Parkinson's and Alzheimer's and so forth. Or we can think about ways of harnessing genetics to at least tilt in our favor or against the favor of a given pathway. So the standard method of developing a drug is this. You think you've got your disease, you get your target selection, you drive through all these phases, and at this point, it's when it gets really expensive. Billions of dollars are spent to go to a phase 3 study, and that's where the most expense is, and it's where most of the failure is. When you ask a drug company why the failure is, if you ask them 30 years ago, the failure was often at this in badly designed studies, and they've learned an awful lot and do very good studies now. The failure is not because of that. The failure is nearly always at this end. And so they've made, the field has made big errors of target selection. And can we do better? Can we drive it? So we can think about Mendelian randomization as our nature's own randomized controlled trial. So as I said before, they're randomly, so RCT is done, you randomize the patients, you give one an intervention, one placebo, the target is engaged if you've picked the right chemistry, and you get your outcome. And if you, and now if you think about the target of those drugs is nearly always encoded by a gene, and that there'll be variation in that gene which will reflect the impact of the target. And I'll give an example of that. So here we've got a compound, it's binding to a target protein, it's encoded by a gene. Of course, the classical one would be a statin, which many people maybe in this room are even on, and you have this huge impact on disease, and your statins lower cholesterol by 1 millimole per litre, cardiovascular risk 25%. Here you've got lowering LDL cholesterol 0.07, coronary risk resistance 6%. But this is based on genetics. So the variants that mimic the action of the statin do modest effects. But you've got much larger numbers for free because these have been genotyped already. So you don't have to do your RCT. You can just go to a genotypes of these patients and say, can you see the same effect? And the answer is, we would have discovered statins using this methodology. And what I want to finish on now is maybe we can think about trying to do that for all the drugs that have been around the cancer field. Some of them have been binned because they do the opposite of what is required for cancer. Maybe some of those may have traction in Parkinson's or Alzheimer's and other neural diseases, if it's right that we are looking at the other sides of the coin. But we can certainly ask the systematic question of saying, can we engage this methodology to look at the targets, because those targets of those proteins or the biologics are known. We know the DNA variation within them, and can we use them to predict the effectiveness of treatments? Has it worked? Yes, it has. This is an example from rheumatoid arthritis, well-known monoclonal antibody binding to IL-6R with this phenotype, sort of fingerprint, and here you can affect on cardiovascular disease again, big impact with the same fingerprint based on the presence of that SNP, which is mimicking the action of that drug, if you like, in a more modest way. And so I think this holds great promise for really short-circuiting that pathway from basic discovery through to therapies. So finally, so I think the evidence about the interplay is still circumstantial. It's looking less circumstantial as the months go by, and I have to say there's a lot of interest, and I was delighted this meeting was being put forward as a result of that. I've been struck both by the two talks earlier today, and I'm sure this will come out in others as the day goes on, is we often speak a different language as fields, and I think we really need to think about how we combine approaches and combine thoughts to really motivate further discoveries. Thanks very much.
We have time for a few questions.
I have a rather naive question. Do you think epidemiologically there are certain population groups or demographics that have less risk of developing Parkinson's?
It's a good question. Well, there's some population groups have an increased risk of feeling Parkinson's disease, so you'd have to say if you took them as your norm, the rest of us have a lower risk. So Jains, for example, from parts of India, have an increased risk of Parkinson's disease. So I suspect the answer is yes to that. I don't think it's massively variable. I think you do see Parkinson's and Alzheimer's, for that matter, in all ethnic groups. So I think it's not going to be a big player, but yeah, I'm sure that's right.
And is that also true for cancer, other than melanomas?
You know, I don't know. When I was in medical school, which was a while ago, you know, there's certain cancers are certain commoner in certain populations for sure. I mean, I remember getting told gastric cancer was commoner in Japanese populations. I assume that's still the case. Anyone working gastric cancer? I think that's still the case. Colon cancer is commoner in people where we eat a lot of red meat. So, you know, I'm sure there are things like that true.
I guess the question I had was whether we could use that information to narrow down correlations between these conditions, right?
Yeah. No, yeah, I mean, it would be nice. I mean, so epidemiological studies, I say I'm a fan of them when they're done really well because they give useful insights. And what you need, I think, with something like that is to transplant people from low risk to high risk and move them around. And you're not allowed to do that. Even your president isn't allowed to do that. Yeah. I'll stop making jokes. I will not make any more any jokes. It's too serious. It's going to be the best. All right. Next question.
Is sun exposure a risk factor for Parkinson's disease?
You know, I don't think anybody's answered that. No, not that I know of. I don't think anybody's looked at it. You think about the sort of melanoma type stuff. Yeah, I don't know. I don't think anybody's looked at it. Do you know if anybody's looked at sun exposure and PD or other risks?
People have looked at the genes that like related to hair color, pigmentation,
And they've not shown up in the GWAS of Parkinson's as a sort of surrogate of that. If we look at people who've got ginger genes or whatever, or freckly genes, they don't seem to have the variants that predict that phenotype, physical phenotype, don't predict Parkinson's. So I suspect not, but.
Sorry. I think, you know, both your comments and the previous speaker's comments about the sort of the difficulty of crossing disease disciplines is I think is a very, you know, important statement. Do you have any ideas for how one kind of shortcuts that? How you get the cancer ideas beyond the neurologists and vice versa? I mean, I've run into this in my career too. Over time, because biology is just biology. The cells don't really know what they're doing or anything like that. And yet, I think, you know, there are things that we can learn. Like, you know, with cancer, you commented about melanoma and Parkinson's. Melanoma is in the skin, Parkinson's is in the brain, but they're both part of the early same tissue, right?
Yes.
And that may be where the link is. And at least in cancer, you get the tissue, you can study the biology a little easier. If you know what it is you're looking at, it maybe tells you more about Parkinson's, even if you're looking at skin. So do you have any insights into what one can do to push that interdisciplinary idea through? Because I think it's a problem.
Absolutely, I think it is. And I think I would hope it would be really great if one of the outcomes from a meeting like this is it's a start of conversations. I was interested by the first speakers, you know, here's a physicist engaging with a whole bunch of biologists and things. And that's it. I think it's trust. You know, you've got to be able to trust the other person and engage. You've got to be able to speak the same language. And I don't just mean English. I mean a sort of biological language. You've got to be sensitive to the limitations. So I went to a meeting that was quite a lot of cancer in it at the Crick Institute in London a few weeks ago, you know, and they're miles ahead of, not just the Crick, I mean biology, cancer biology generally, are miles ahead of where neurodegeneration is in lots of ways. But then I read some papers of cancer where they talk about mitochondrial biology in cancer, and it's quite primitive compared to where neurosciences are appreciations of mitochondrial dysfunction, because we've been around mitochondrial dysfunction. So there are lessons to be learned. I don't have a magic solution. I think it is going to come down to personal relationships where people say, I really need a certain type of biologist to help me with this, or a certain type of genomicist or big data person, and but I think they'll start with conversations.
We have time for one last question.
Great. So I thought that your approach to the polyglutamine diseases was really interesting, and it's based on the notion that there's a common underlying biology for those sets of conditions. A lot of people in the neurodegenerative field think that there's common biology between Parkinson's, Huntington's, CTE, the tauopathies, the synucleinopathies, and all of these different things. Has a similar approach to risk alleles for protein misaggregation or those sorts of processes been taken across diseases?
So, not for some of the diseases you mentioned, because of the asymmetry in the population sizes. So for Parkinson's, we can get to 15,000 more cases now. I should have said there's going to be another paper coming out in the next few months, I think, with more genes coming out. With Huntington's will be 2,000 or 3,000. So there's an asymmetry in the sizes. There was, when we did the Parkinson's GWAS some 6-7 years ago, we talked to the Alzheimer's guys, and I would have bet, not a lot of money, but a bird amount, saying there'll be some commonality, there'll be some risk factors which increase your risk of neurodegeneration, and some that don't. And actually, there seems to be virtually no overlap between the two diseases, which was a bit of a surprise to me. I would have thought biologically you'd expect a few alleles. Now, it could be then we were talking maybe of just having maybe 2,000 or 3,000 Parkinson's cases and maybe 4,000 or 5,000 Alzheimer's. Maybe we do a better job if we have another look. So people are looking across two big neurodegenerative diseases. People looked at ALS and Parkinson's, not found very much. Of course, ALS and overlap with frontotemporal dementia is a well-known thing in terms of Mendelian forms of disease, but I haven't seen a GWAS showing interactions yet. But I think you just have to keep your eyes open, really. Keep one's eyes open.
Thank you so much.